Good morning, students. Good morning, everyone. Good morning. Am I audible? Am I audible? Okay. Sajad, please ask Subham to look into that matter. Actually, they have a lot of details, professional bodies.
मैम मैंने आपको एक फॉरवर्ड किए मेल आप जरा देखिए प्रोफेसर बॉडीज आप उसमें जो आप बता सकते हो भर दीजिए
Year of renewal, establishment, duration of membership, name of faculty member, list, attached list, everything. If I put not applicable, then 1 second they will keep on sending. Yes. What is the last class we have discussed?
आप नीचे क्या लिखते हैं ये लिख दिया है ना
Yes, toxin we have discussed. Okay. Bacterial toxin and types. Okay. Is there any doubt in that previous classes? Is there any doubt in that previous classes? No. Okay.
हाँ जेएमडी एएचएमडी डीएचपीटी का डीएचएटी का
So today we are going to discuss about bacterial spore production. Bacterial spore production.
एसएजेडी
The slide is visible to you?
E10675
Okay. You just tell me the slide is moving or not. Slide is moving. The slide is moving. Right. So what is spore? What is spore? See, a number of particularly gram-positive bacteria, the number of gram-positive bacteria form a special resistant dormant structure. That structure is called endospore. Okay, so this type of endospore will commonly found in gram-positive bacteria. So mostly gram-positive, number of gram-positive bacteria form a special resistant structure is called the endospore. So these endospores develop within the bacterial cell. It's not developed from outside of the cell or somewhere in the environment. It is developed within the bacterial cell. Take for example in Bacillus, Clostridium. So these are the bacteria which produces spores. So these structures are resistant to heat, ultraviolet radiation, gamma radiation, chemical disinfectant, and desiccation. In fact, some endospores remain viable for even after 1 lakh years, and actinomycete spores have been recovered alive after burial in the mud for 7,500 years. So these kind of endospores will be viable for even 1 lakh years, and the actinomycete spores have been recovered alive after 7,500 years. Because of their resistance and the fact that several species of endospore forming bacteria are dangerous pathogens. So those bacteria which form spores are dangerous pathogen. The endospores are of great practical importance in food, industrial and medical microbiology. So these kind of spores are produced only during unfavorable condition, when there is an excess of heat, high temperature, that is heat, temperature, pH, alternation in the pH, or due to the lack of nutrients, ice salt concentration. All these things are the unfavorable condition for a bacterial growth. So in order to escape from these unfavorable condition, a bacteria produce will produce spores. The spores may be, you can see in the picture, it may be centrally located. The spores may be centrally located. The spores may be centrally located, or sometimes close to the one terminal end. It's not completely to the terminal, close to the terminal end, that is called subterminal, or sometimes it will be completely terminal. Now we come to the structure of spore. See, this is the spore. Inside this is the spore. The core is nothing but the spore. The spore is surrounded by a, so just we can start from here. The spore is surrounded by a thin, delicate covering. So this is a delicate covering outer side. Thin, delicate covering called exosporium. Under this exosporium, a spore coat. So this is exosporium. Under this exosporium, a spore coat is there. It is composed of several protein layers. The spore coat is composed of several protein layers, and the spore coat is always thick, fairly thick. It is impermeable also. That means no material can easily penetrate through that. It is impermeable. No substance can be penetrated through the spore coat. So it is impermeable and it is resistant to chemicals. So no chemical make any harm to that spore coat. So it is resistant to chemicals. Then under this spore coat, the cortex is there. So this space like this is a space, the cortex. The cortex occupy half of the spore volume. You can see half of the spore volume is occupied by the cortex. Actually, the cortex is half of the spore volume. Then there will be another spore coat, right? So half of the volume was covered by the cortex. It is fully composed of, this cortex is fully composed of peptidoglycan. It is made up of peptidoglycan. Then under this cortex, a spore cell wall is there. Under the cortex is the spore cell wall, that is the germ cell wall in the picture. It is a spore cell wall or core wall inside the cortex, and it surrounds the protoplasm or core. It surrounds the spore. Then 15% of the spore's dry weight consists of dipichloric acid. So 15% of the dry weights, spore's dry weight consists of dipichloric acid, and this dipichloric acid is nothing but it is a complex of calcium ions which is present in the spore. Now coming to the sporulation process. The process of spore formation is termed as sporogenesis or sporulation. Sporulation is nothing but the synthesis of spore from the vegetative cell. The process of formation of spore from the vegetative cell is the sporogenesis or sporulation. So normally this kind of sporulation occurs when the growth stops due to lack of nutrients or due to unfavorable environmental conditions like pH, temperature. As I already told you, when there is an unfavorable condition, in such condition the bacterial growth cannot be possible. The bacteria struggles for, if the bacteria struggles for its growth, then in order to escape from these type of unfavorable condition, in order to escape from this unfavorable environmental condition, the bacteria forms a spore, and that process is called a sporulation, right? So how the environmental conditions occur? The unfavorable environmental condition happens due to the lack of nutrients or the elevation or decrease of temperature. or the elevation or decrease of pH. So it is a complex process. The sporulation is a complex process. It may be divided into 7 stages. The process happens in 7 stages. First, stage 1, the genetic material, that is the DNA of the bacterium. Inside that, the first stage, the DNA of the bacterium is duplicated. The 4 spore begins to isolate the genetic material of the bacterium is duplicated. That is, the DNA of the bacterium is made itself a double copy. The 4 spore begins to isolate the newly replicated DNA and a small portion of the cytoplasm.
From when you are facing this problem?
Then second stage, invert folding of the plasma membrane starts to surround the replicated DNA and cytoplasm to produce the 4 spore, and that is called septum. This smaller cell is called the daughter cell. The remainder of the bacterium is called the mother cell. Next, we will come to the third stage. This membrane continues to grow and engulf the immature 4 spore in the second membrane. In the fourth stage, the peptoglycan forms between the two membranes. Next, cortex is laid down in the space between the two membranes, and both calcium and dipicolinic acid are accumulated. Stage 5, the protein coats then are formed around the cortex. The new spore loses its water and becomes highly dehydrated to that point. All remain in the spore will be. So after that, after dehydration, the remaining all remains in the spore is the DNA, small amount of RNA, ribosome, enzymes, and some important molecules. Then in the sixth stage, maturation of the spore. In seventh stage, the endospore is released from the cell and it can remain dormant for even 1,000 years. So the formation, the sporulation process requires only 10 hours in bacteria. I will show you a video clip file how the sporulation process happens.
Dr. Godkar, Ramnik, Sood, Panikar, and what else book we have decided for the in this MLT?
dormant heat and desiccation resistant spores. Most of our knowledge of bacterial sporulation comes from the gram-positive soil bacterium, Bacillus subtilis.
Yes, you can
When growing
see
in rich
in the
media,
picture,
this microbe undergoes normal
this is the
vegetative growth.
normal
B
bacterial cell.
subtilis can
Now
replicate
the
every
DNA
30 to 60
is
minutes.
doubled, that is duplicated. The DNA is make copy
In
itself
contrast,
into 2 DNA.
starvation
So
initiates sporulation,
what
an
happens?
elaborate 8-hour genetic
This is the first
program
step.
that ultimately
The
yields a
first step,
spore.
the cell
Sporulation can be divided into
make
8
copies of its
morphological
DNA.
stages. Stage
That is the first
0
step.
represents the point at which the vegetative cell decides to
And
use one of
then
2 potential polar division sites to begin
one
septum
portion,
formation,
a small
instead of the
portion.
central division site used for vegetative growth. Stage 1 involves replicating
A
and stretching
small
the
portion of the
DNA into a
DNA.
long axial filament that spans the
Yes, a
length of the
small
cell.
portion of the DNA
Ultimately,
is
one of the
just
polar division sites
departed.
wins out, and in stage 2, septation
That is a
occurs,
septum.
dividing
The
the
small
cell
portion
into
of the
2
DNA
unequal
gets separated.
compartments. The forespore, which will
Small portion of
ultimately
that is
become the
called the
spore,
forespore.
and the larger mother cell from which it's
And this one is
derived.
the mother cell,
Each
and this one is the
compartment
daughter
contains
cell.
a chromosome.
This is the mother cell, and this is the
In
daughter
stage
cell.
3 of sporulation, the mother cell membrane
Then what
engulfs the
happens?
forespore,
The mother
resulting in a
cell
structure
engulfs the
encircled
daughter
by
cell.
2
Just see.
membranes. In stage 4, the mother
The
cell
mother cell
chromosome is destroyed,
engulfs
and
the
a
daughter cell,
cortex, made
and
up of
this
a
is
thick
formation of cortex.
peptidoglycan layer, is placed between the
Then
2
formation of
membranes
cortex.
surrounding the
The
forespore
cortex is
protoplast. This
completely filled
cortex
with the
will be important
peptoglycan.
in providing heat resistance
The
and
cortex is
maintaining
completely filled
dormancy
with
to the spore.
peptoglycan.
In stage 5, layers of coat proteins are deposited on the outer membrane. Stage 6 completes the development of
Then
spore resistance
slowly
to heat
dipicolic
and chemical
acid and
insults.
all the cells with
This last stage includes the
all the cells
synthesis of dipicolinic acid in the mother
dipicolic
cell
acid.
and its uptake into the core of the spore.
All these
Dipicolinic
things are
acid
filled
is
up in that.
important in maintaining dormancy and in promoting spore dehydration, a characteristic that makes the spore heat resistant.
Then
In stage 7,
the
the mother
water,
cell,
moisture
now
content which
called a
is present
sporangium,
inside the
releases the mature
cell
spore.
is removed, and
Spores
finally
are
the cell
resistant to
is
many
released.
environmental stresses that would
And this
kill
is
vegetative cells. The nature of this resistance is due in
sporulation,
part to spores
sporogenesis.
having only 10 to 30% of
Clear?
vegetative cells' water content. But spores also contain small acid-soluble proteins that bind to and protect DNA from damage by ultraviolet
Then these
light
kind of
and
spore
various
will be
toxic
remain
chemicals. Fully
viable for
mature
even
spores can
1,000
exist
years,
in soil for at
7,500
least
years.
50 to
When
100
there is a
years and
favorable
have been known to
condition
last
occur,
thousands of years.
it will
Once
break
proper
down
nutrient conditions arise,
the
another
spore coat,
genetic
and it
program
will once
called
again it will become a
germination
normal
is
bacteria.
triggered to wake the dormant cell,
Once again it will
dissolve the
become
spore
normal
cortex
bacteria.
and coat, and release
Once
a
the favorable
rehydrated,
condition occurs, if
viable
there is
vegetative
nutrients,
cell.
if there is normal pH, normal temperature, it once again it become normal vegetative bacterial cell. So that's all about sporogenesis. If you have any doubt on this, you can ask me now.